Hi, my name is Ellie Nichols, and today I'll be discussing the field of epigenetics. Have you ever looked at yourself in the mirror and asked, why am I the way I am? Why do I have the hair color I do? Why am I the height I am? And why do I or don't I look like my parents? Well, all of this can be traced back to your genetic code, which writes out instructions for who you are and how you will change over the course of your life. You've probably heard of genes, sequences of DNA or RNA that code for certain molecules within the human body. Genes, which are specifically ordered sets of nucleotide base pairs from nucleotide bases adenine, cytosine, guanine, and thymine, are found on chromosomes. Normal human beings have 23 pairs of chromosomes, 22 pairs of somatic chromosomes, which code for a variety of genomic and physical traits, and 1 pair of sex chromosomes, which contain information that controls what your gender will be. Often these genes fit a certain physical or chemical standard and are seen as normal by scientists. However, sometimes the genes mutate, which can result in physical or chemical alterations of the body, and they can be passed down throughout the generations. The field of the study of inheritable mutated genes is known as epigenetics. Within the context of epigenetics, genes mutate due to a variety of factors, and often their functions and expression, or how the gene physically manifests itself, change as well. However, here's the twist. While some gene mutations are a result of the change of the genetic sequence, or the base pair ordering, epigenetic changes do not involve any alterations of the actual DNA sequence. Rather, they happen because of modifications of other aspects of the genome. A gene that once coded for one specific function after undergoing mutation could have an altered role in the body or a new one altogether. These changes can change normal chemical functioning in the body even from birth. There are 2 primary mechanisms by which genes can be changed without changing the base pair order: DNA methylation and histone modification. In DNA methylation, a methyl group, 1 carbon and 3 hydrogens, is added onto a DNA base pair, either cytosine or adenine. On cytosine specifically, it replaces a hydrogen molecule. Chemically, this changes the structure of the base, but the base identity remains the same, maintaining the structure of the DNA as it was before. In histone modification, histones, the type of protein that packages and transports DNA, are chemically altered. This can occur through methylation, where methyl groups are added, acetylation, where acetyl groups are added, and more. As before, the actual sequencing of the DNA isn't changed. The gene, however, is fundamentally altered by the addition of the methyl or acetyl group, causing a change in how the gene expresses itself. These processes result in altered gene expressivity, which can be visible to the human eye, or more subtle biochemical modifications. The mutations in the gene result in a wide variety of epigenetic effects. Many times, the result is benign and crucial for the body. For example, epigenetic changes are the reason why skin cells look different than internal organ cells, because there are changes to what is around the DNA, but the DNA within the cell is the same as in other cells and throughout the body. On the other hand, methylation of cell replication control genes can result in a higher amount of cell differentiation or cancer. Epigenetic changes, whether insidious or not, are significant to the body's functioning. This field, far broader than I could hope to describe here, is a truly fascinating area of study that has changed how we understand genetics and will continue to have important impacts in the future as we learn more about it in the fields of medicine, genetic research, and more.